everybody. Why don't we get started so we can give John the maximum amount of time. Thanks for coming to the Rough Cut. I wanted to make a couple of announcements, first of all, about things to come. On Monday, the IOE is having a distinguished visiting lecturer come in. That's going to be Paul Robbins. He's director of the Nichols Institute at University of Wyoming. He's a political ecologist, so he's going to be talking about some of the social dimensions of conservation and his work in India and Montana and other places. So that's 7 o'clock at Museum of the Rockies. Hope you all can make it. The other thing is we are having a Rough Cut next Wednesday, kind of a suddenly scheduled one. We're having the executive director of the Global Change Research Program talking to us, coming from the White House. He works for the Office of Science and Technology. He's going to be coming in, and he'll be giving a Rough Cut same time, but it'll be in the Procrastinator Theater. So his name is Tom Armstrong. So we're very excited about that too. But we're very excited about today as well. I'm happy to introduce John Peters, professor of chemistry and biochemistry, who's been over the years director and leading a bunch of really important initiatives on campus, from thermal biology to astrobiology, and now this new Energy Frontier Research Center at MSU called BETSI, which he's going to talk about today. This is one of 32 centers selected from around the country. John's lab works on enzyme structure and function, microbial physiology and ecology, evolutionary biology, and bioenergy and biotechnology. So I imagine in the next hour we're going to hear something about all of those. So thank you, John, for coming.
Yeah, thanks, Kathy. Today what I've tried to do is kind of bring together some new results we have that I think are kind of exciting, but also in the context of some new bigger projects. We have multi-PI projects, which I think are pretty exciting too. I used a new background, and so anybody but Rob, guess what this is? Baldy. Yeah, it's Baldy, and it's coming. I got the opportunity this summer to work the sweep with Rob Maher, and actually Rob took this. I didn't know Rob was going to be here, but Rob took this picture of me ascending to the last aid station in the ridge run. So I'm using that as a background. I wasn't going to make a big deal about it, but since I saw you, you're out. Sorry, I didn't know. Okay, so the current research in my lab, we've kind of shifted over the last year. So we had this research in astrobiology. That was a big emphasis in my lab. We had a big center funded from the NASA Astrobiology Institute to look at enzymes and specifically iron sulfur enzymes in the context of the origin of life. And so, those enzymes, they're really important enzymes in kind of simpler metabolism. We work on enzymes. I'm going to talk about this in more detail, hydrogenase enzymes. So enzymes that work reversible hydrogen oxidation, which we think was important in early metabolism. But it's really also important for biofuels, and hydrogen itself is an important potential biofuel. So, the same sort of problems, the energy problems that life had in the beginning to get going, we still have those sort of problems. And the unifying feature to those is really the control of electron transfer. So the control of the flow of electrons in metabolism, and that's really one of the things my group is primarily interested in. So we have this new biofuel project that Kathy mentioned, the BETSI Center. So this is Biological Electron Transfer and Catalysis. This is a DOE Energy Frontiers Research Center, and so we're fortunate to be able to lead this center from Montana State University, but it has there's 11 PIs on the project. It includes 5 universities and the National Renewable Energy Lab. And I'll talk a little bit more about what the emphasis of that center is in terms of our research. Another multi-investigator project that we just got started last year is in agricultural biotechnology, and so in nitrogen fixation. And so this came out of a solicitation that started with a workshop, but it was a workshop under the auspices of lowering the inputs of nitrogenous fertilizers in agriculture, so mechanisms to do that. So this was a new mechanism, a new funding mechanism that NSF has been playing with, called this Ideas Lab. And so what they do is you apply for a workshop. They pick not just on the basis of your scientific credentials, but you write stuff, and it's a little bit of a psychological or sociological kind of evaluation of you, and then they throw you all together. And this was 15 people from the U.S. and 15 people from the UK. You do a bunch of cheesy icebreakers over the first couple days, and then they say, okay, well, there's $12 million, and you guys need to develop independently proposals and then fight over this money and evaluate each other's proposals over the course of a week. And so this is a mechanism that NSF is playing with to get more innovative research. And so we were fortunate to lead one of those, and this is another multi-investigator project that NSF and BBSRC, so we're the lead PI in that project, and this guy Phil Poole at Oxford is the lead PI of the UK portion of that. So those are two big areas that we're working on in my lab now. Another one is also electron transfer centric. We're trying to exploit our knowledge of electron transfer reactions to develop bio-based chemical sensors based on redox reactions. And so for a good sensor, you need something that has a high degree of specificity, and you need an amplifiable signal. And so our idea is we'll use the billions of years of evolution for the sensitivity, for the specificity. And enzymes, have high specificity for their substrates. They use redox reactions that do oxidation reduction, and then couple that to either attachment to an electrode, which is amplifiable, or various dye reagents or fluorescent reagents. And so one of our models there is an enzyme Mercuric Reductase, and so that converts mercury 2 to mercury 0. It's involved in environments where there's large amounts of mercury. Microorganisms can flourish there. They detoxify themselves from the deleterious effects of mercury. Mercury 2 readily modifies styles and mucks up your biology, but they have this enzyme that will reduce it to mercury 0, which is more volatile, then it goes away, at least away from them. So we're looking at enzymes from hypothermophile, because these have a lot longer shelf life. They're stable. We can treat them sort of like materials. And so the idea here is to make a material from these stable enzymes and couple redox reactions to detect things like mercury. Another project in the lab, and this is really the project that's funded by IOE and EPSCoR, is Amaya Garcia-Costas in the lab is working in this environment in hailstone. And so we have, over the maybe past 5 or 10 years, have had programs looking at life in extreme environments and really defining sort of the parameters or what constrains different biochemical processes, like what environmental parameters constrain the occurrence of nitrogen fixation or aspects of hydrogen metabolism, different aspects of metabolism. And we've worked in Yellowstone some, in the Great Salt Lake. We've had for several years now an AFOSR, an Air Force funded project to look at the ecology of different biofuel producing organisms in the Great Salt Lake. This hailstone is another really hypersaline environment that Amaya is working on. So that, I'm not talking about that today, but this would be a really nice topic for a future one of these, where Amaya, who's actually the intellectual driving force for this project, could lead. So down to business here, and I know that this crowd is a little bit of a diverse crowd, so I apologize if I'm giving you sort of a rudimentary metabolism lesson, but but it'll go by quick, and it's a good basis, a starting point for the things I'm going to talk about. And so, when I'm talking to, I teach organic chemistry and biochemistry to in the pre-nursing program, so at the 100 level. And when we start to talk about metabolism, we break it up into catabolism and anabolism. Catabolism is breaking down things. It's oxidative processes that are energy yielding, and anabolism, the other arm where we're building things up, and these are reductive processes that are energy requiring. And we talk about metabolism in just 4 steps. I like to talk about it in 4 steps, which are basically 4 conversions between different forms of energy. You have reducing potential in like carbohydrates or fats or proteins. That reducing potential, what we do in metabolism is oxidize stuff. And when we oxidize stuff, we convert that reducing potential to this unifying currency of reducing equivalents, and that's really NADH here, okay? So anytime you oxidize something, we oxidize a carbohydrate, we can reduce something like NAD to NADH, and then we've got this kind of unifying currency. It's like the euro, but even the British have to participate. Everybody has to participate in this, at least to some extent. I'm going to talk about some examples where certain aspects of the biochemistry that we really normally associate with NADH, some microbes can do that without NADH, and that's part of one of the topics I'm going to talk about. So oxidation reduction reactions, that's kind of the first step, or the first couple steps in this conversion between these different energy forms. We've got reducing potential, like in carbohydrates, reducing equivalents, this unifying currency like NADH or FADH2 is another one. Those are used to power this machine in an aerobic metabolism and respiration, the electron transport chain, which then that converts the reducing equivalents to a proton gradient that then can be converted to chemical energy, the 4th type of energy in the form of ATP by ATPase. Okay? So that's aerobic metabolism. Anaerobic metabolism is still sort of oxidative, and this is glycolysis, and so I'm going to talk about glycolysis in detail. But really what glycolysis is doing, it produces ATP by substrate level phosphorylation. That's really what we call the energy conservation mechanism for anaerobic metabolism. Substrate level phosphorylation, and then we make NADH. So NADH, I just talked about that as one of the steps in like an aerobic metabolism, getting a lot of energy. That's great if you can do respiration, not necessarily aerobic respiration, but if you can do respiration, then NADH is good stuff, and we can actually get more energy out of that, like electron transport chain, proton gradient, ATPase. In anaerobic metabolism, so that really is the aerobic fate. If we have oxygen around, and this is when we talk about basic metabolism, we talk about the 2 fates of pyruvate in metabolism. The aerobic fate goes to acetyl-CoA, that goes to the TCA cycle. Then you make a whole bunch more NADH and can make more ATP. The anaerobic fate really has to solve a problem in anaerobic metabolism because the NADH no longer is kind of a boon that you can make more energy out of. We have to be able to recycle this back to NAD. So if you have a pool of NAD, you tie it all up in NADH as you're oxidizing glucose in this, then that becomes a problem. So you have to re-oxidize that. So that's this other anaerobic, an example of an anaerobic fate of pyruvate is the conversion of lactate by lactate dehydrogenase, and in that you reduce pyruvate to lactate with the NADH and recycle the NAD so you can do more glycolysis, okay? So this, if you know all this metabolism, then just bear with me for a second, but I need this for the basis here. So like I said, in aerobic metabolism or in respiration in general, it doesn't necessarily have to be aerobic. There's other terminal electron acceptors for electron transport chain, like nitrate or fumarate, where you can do respiration aerobically. If you can do respiration, use the electron transport chain to make more ATP. The pyruvate converted to acetyl-CoA is funneled into the citric acid cycle. And then the electron transport chain. And so this is really the second mechanism we talk about for energy conservation. So we've got substrate level phosphorylation. That was represented in these reactions, and I didn't really say anything about them, but I can go back. These reactions here where we transfer a phosphate from a metabolic intermediate to ADP to form ATP. That mechanism is called substrate level phosphorylation. So that's really based on generating these high energy phosphate containing intermediates in metabolism. Okay? And then this other mechanism, if we have an electron transport chain, and we use the electron transport chain to make a proton gradient, that proton gradient drives this ATPase, and we have a different reaction, ADP plus inorganic phosphates make ATP. That's electron transport phosphorylation, and that's a different and the second mechanism when we talk about energy conservation mechanisms. I'm setting it up in this way because I'm going to talk about one of the thrusts of our BESI project is a third mechanism, or now what we think is a third mechanism, energy conservation, called electron bifurcation. Okay, so some simple forms of life can do this kind of metabolism, like fermentation, respiration, without, I said this was the unifying currency. Well, you don't necessarily have to use NADH to do some of the same metabolism, and you do that sort of via hydrogenase enzymes. And so, you know, hydrogen itself is kind of what we think about, it might be a kind of a primordial form of NADH. It's just like NADH without all the decoration. You got the electrons and the hydrogens, but you don't have the big cofactor, okay? So we think that that may be sort of a simpler start for this type of metabolism. And really, this is represented in a couple examples that I'm going to talk about. These are pretty amazing enzymes. They have really funky metal clusters, okay? And so that's what I'm showing here. So this is one we call an iron hydrogenase and nickel iron hydrogenases. They're just really named because of their respective metal content. But they have these enzymes that have these active site clusters, or where we actually get reversible hydrogen oxidation. This reaction to occur is at a metal site that has carbon monoxide and cyanide ligands. So normally, things that we associate with a high degree of toxicity, these are part of the active enzymes in the case of this. This is the iron center here. There's two irons. It seems kind of dumb, iron center, but this one was here first. In this case, this is a hetero bimetallic site, and it has two metals, so it's like nickel iron, and then, okay, well, what do we call this one? We'll call it iron, even though it sounds stupid, but it's just because of the perspective of this. So this is really the only place in biology we see these strange ligands, these carbon monoxide and cyanide ligands as associated with this reversible hydrogen oxidation reaction. And these two enzymes, they occur in different forms of biology. We have iron hydrogenases that are found in bacteria in lower eukaryotes. Maybe in archaea, there's a couple reports, but I'm not sure I'm believing that. And then in nickel irons, for example, there's no iron irons found in cyanobacteria, no nickel irons found in lower eukaryotes. So their occurrence is different, even though they share this sort of ligand architecture, and they're evolutionarily unrelated. And the most closely related evolutionary protein to an iron hydrogenase is this protein called NAR1 that's only found in eukaryotes. It's involved in cytoplasmic iron-sulfur cluster biosynthesis. And then this nickel iron, they're most closely related to respiratory complex 1. And this is something that I'm going to focus on. But this is pretty amazing, and I refer to this as at least the most profound case of convergent evolution, because we had two paths that funneled in onto this really unique architecture. These things, even really fascinating mechanisms to build these ligands in both of these proteins, but they're completely unrelated. No aspect is really shared between the way biology makes and inserts it makes these metal clusters, which is, I think, just really amazing. Okay. So via these hydrogenase enzymes, for example, this is sugar metabolism in this microorganism Clostridium pasturionum. Clostridium pasturionum, it ferments sugars to make organic acids. It makes acetate. It can make butyrate, which we, for a long time, grew these microorganisms in the lab, grew them in the chemistry building. We got a lot of flack for that because they can make butyrate as the end product. And I tell you what, a little bit of butyrate goes a long way. This stuff is just like pooey diaper smell that can just penetrate an entire building. But you can see, you know, when Clostridium pasturionum, it ferments glucose to acetate, it's oxidizing glucose. It produces reducing equivalents in the form of ferredoxin. So ferredoxin is just really simple iron sulfur protein, has these cubes of iron and sulfur. This was coined and discovered by this guy, Lynn Mortensen. He was at Purdue University at the time. So it's just kind of a conduit that then takes those electrons from glucose oxidation and then reduces this hydrogenase. And so it's recycling these ferredoxins via the production of hydrogen. That's what the CP1 does. So making this instead of NADH, but we couldn't oxidize more glucose unless we recycled this back to the oxidized state. And that happens via this hydrogenase. So this is called CP1, so it's Clostridium pasturionum hydrogenase 1. And there's more than one hydrogenase there, which I'll talk about. So. This is another example, and this is on the respiration side, okay? So it's sort of an analog to what I said, the aerobic metabolism or respiration in general, and this is sugar metabolism of Pyrococcus furiosus. And so it uses a membrane-bound hydrogenase complex to create a cation gradient, a proton or a sodium gradient that then can be used by an ATPase to make ATP, sort of analogous to respiration. And this is really stuff that's been pioneered by Mike Adams at the University of Georgia, and he's a member of our BETSI team. This is some work that was done by Eric Boyd in my lab. Eric Boyd was a postdoc in my lab. Now he's a junior faculty in the microbiology department here, microbiology and immunology department. So he's really looking at the phylogeny or classifications, different clades of nickel iron hydrogenases. And so if we go back to those examples, this is an example of a role of a iron-iron hydrogenase. And in Mike's work, this is a nickel iron hydrogenase. If we look at the different, nickel iron hydrogenases are more diverse than the iron-iron hydrogenases. They really, if we do this sort of unrooted phylogenetic representation, they break off into really what we can say is three major clades. There's the group 1, which is the structure that I showed you, and this is from sulfate-reducing bacteria. Sulfate-reducing bacteria use oxidized hydrogen and reduce sulfate. And you have group 4 here that I'm going to focus on first. Group 4 are what I just showed you, this membrane-bound hydrogenase. And actually, if we look at this evolutionary path, this is on a path to NUO. Anybody know what NUO is? NADH, ubiquinone, oxidoreductase. Everybody know what that is? That's complex 1 of the respiratory chain. Okay, takes NADH, oxidizes NADH, reduces ubiquinone. That's the first step in that, and drives proton pumping, makes ATP. If we look closer here, we can make an argument that these membrane-bound hydrogenases are the evolutionary ancestor of NUO, or ancestor of complex 1. Okay, so these are archaeal enzymes. Archaeal MBH, or this membrane-bound hydrogenase from Pyrococcus furiosus, is the ancestor of the modern respiratory complex 1. Okay.
Does that one actually interact with ubiquinone? That we
Yeah, NuO is the complex one, and the MBH does not. Okay, and that's, I'm going to talk about that. Okay, I'm focusing in on some new results we have that will address that directly. Okay, so this is a really complicated complex here that instead of taking electrons from NADH and transferring to ubiquinone while powering this pump, it takes electrons from a reduced ferredoxin. So this iron sulfur protein, same thing I showed you in the Clostridium metabolism, and then reduces protons to hydrogen, and in that process powers this proton pump. So we can get a proton gradient, make ATP, proton or sodium gradient, and make ATP via a ATPase. Okay? Large 14 subunits, and 14 subunits that are analogous to subunits in NuO, or now I've got NQO, so NuO, ubiquinone, and NQO is just quinone. They're the same beast, essentially. And you can see that most of these subunits, or a lot of these subunits, are analogous. We've got a little bit more membrane components here in the Pyrococcus furiosus hydrogenase. We've got a whole suite of cytoplasmic enzyme exposed subunits that really make this possible. Okay, so we really got to think about this like as a protein complex that's instead of going from NADH to reduced quinone, from ferredoxin to reduce protons or hydrogen. So it's just transferring electrons down this electrochemical potential, and through that the energy's harnessed to run these proton pumps. So these enzymes do it different ways. In respiration, we know complex 1, NADH, to quinone or ubiquinone. Here, the MBH, ferredoxin, to end up from hydrogen. Okay? So the structure of NuO has been determined from Thermus thermophilus. And fortunately, this is enough related to our system that we can kind of use this as a starting place. So this is supposed to be the membrane, this gray part, and these are like membrane spanning helices that attach it to the membrane. This has the proton pumping capability, and this is really the machine where the electron transfer happens that can power this proton pumping. And so you've got a diaphrase part, or diaphrase is this functionality, the NADH oxidation, and then you've got this quinone reduction, which is close to the membrane. Quinone kind of hangs out in the membrane, but actually this site for quinone reduction in this is sort of a little bit outside here. But you can see, you know, the coenzyme Q, it has this long, greasy part that can, and presumably that's directed to a quinone site. So some of the things we're really interested in is like, how do you evolve? You know, if we think about our protein as an evolutionary ancestor, or Mike's protein, I should say, really this MBH as the evolutionary ancestor of complex 1, how did it make this transition from, instead of oxidizing ferredoxin, oxidizing NADH? And even more interesting, how did it make, interesting for us anyway, how did we evolve from this complicated site that I told you about, this nickel-iron site that has this nickel and iron and COs and cyanide? How did it evolve from that site, which reduces protons to hydrogen, to a site that would bind quinone and reduce quinone to quinol? Okay? Okay. First thing that Mike wanted to ask is, do they form these similar complexes? So this was determined, this structure was determined and published in Nature. I showed you that. We didn't do that. These guys did that. Mike wanted to ask, well, does it form the same sort of complex in the membrane? And they did that by small angle X-ray scattering studies, by this guy's a real character, John Tainer at LBNL. You cross paths with him? Oof. Double character. Yeah, but he's good, and he does some great stuff. So he did these small angle solution X-ray scattering where you can just look at the shape of molecules in solution. And so they asked the question, does what we get out of the solution X-ray scattering studies, which is blobby stuff like this, does this look like that? And the conclusion was that, yeah, it sort of looks like that. So it's probably forming a similar complex. We've worked with Mike to try to crystallize, and we do protein structures in my lab, and that's one of the things that we do as a whole suite of tools we use. It seems to be the one that just gives us the power. This is like where we get our science and nature papers. Everybody loves these structures, and when you can look at a structure as a biochemist, you can ask a ton of nice questions, scientific questions. So everybody loves these structures, and they still love the structures. So we still try to do some structures. So Oleg in my lab went about trying to crystallize this MBH from Mike Adams' lab. And so they went through a number of trials of ways to express it. So, solving membrane structures is just a mess, because you've got like a part that doesn't want to interact with water and interact with a membrane, and a part that does want to interact with water. And so trying to get that to set up in a crystal, in a regular crystalline array, is sometimes a real challenge. So what we did here was just tried to do it in a way where we got rid of most of that membrane stuff. We were more interested in these questions associated with NADH, associated with quinone, so we got rid of that part. And so Mike figured out a way to just, in Pyrococcus, he developed the genetic system and a protein expression system in Pyrococcus. And then in addition to that MBH enzyme, which they need for the Pyrococcus metabolism, he just used another construct to express more of these subunits that kind of hung out in the cytoplasm, okay? So that's N, K, L, and J, where you had another construct where you had one of the membrane anchors and N, K, L, and J. And so after a lot of trials, Oleg finally got crystals of, actually got crystals in what Mike calls MBH1, which he said was the tetramer. He got crystals of the pentamer in that, but it has a little bit of contamination of this other M subunit in what he's calling the tetramer here, so we selectively, and that's the way this crystallization stuff works. There's just, even after the many years of being involved in this stuff and asking how we can sort of treat this as kind of a scientific, methodical thing, it's just like throwing a bunch of stuff at it and hoping for the best. And so the more stuff you throw at it, the more things you try. And so Oleg tried a whole bunch of things, and that's what he got to work. He got some crystals that diffracted to 3.3 angstrom resolution. That's not very good resolution, but what we're really interested in is the overall architecture here. And those are the type, that's all we need to really answer the key questions, at least initially. And so this is the structure that he solved. He solved this N, J, L, K, and M. And so this has iron sulfur clusters, and it has the site that interacts with the ferredoxin. And then this L and K part is what has the nickel ion hydroxide. So I'll kind of take you through some of the highlights of that, not all the details of the structure because of time, but just kind of the highlights. Here's, we're showing it embedded in the membrane. We've got sort of a problem here because usually these helices, they kind of line up perpendicular to the membrane, and so we either have something artifactual going on with our M subunit or our anchor, or it has a kind of different interaction in the membrane. We don't know yet. Fortunately, this isn't where the important questions are, and so really the funneling in on, I think the evolution of the Q site is really the key interesting thing here. So this is looking at it side by side, again reminding you the major differences here, or the similarities. And amazingly, we have this same sort of array of iron sulfur clusters, and reminding you what this thing does. It like takes electrons from NADH and then passes them like a bucket brigade to different partners, and through that, going down an electrochemical gradient, it harnesses that energy to pump protons, okay? And so this is the bucket brigade here. Going from NADH, the diaphrase here, and then here to the quinone, okay? Through that, you power to pump protons, which then you can make ATP from. And so we've got just a portion of that array here. We don't have this complicated diaphrase. We're just using electrons from paradox. And so iron sulfur cluster to iron sulfur cluster. And in fact, the iron sulfur clusters associated with this paradoxin are these kind of cubey things that find all over the place in biology. Okay? So that's conserved, and then what we see is that at the site of complex 1, where quinone is reduced to quinol here, that's the site of where protons are reduced to hydrogen. So really we're looking at the evolution of a quinol site happens via this complicated metal center. So that's a surprise to me that you have cast off this metal center to make an open site that binds quinol.
John, how efficient is that conversion of hydrogen to proton pumping?
Yeah, I don't think Mike has, and that's a difficult thing to measure, but I don't think they've measured how. And that's an interesting question. Does it work as efficiently as a complex 1? And we don't know that. Yeah.
Is there any sequence homology around that site, or is it just a...
Yeah, that's what I'm getting to. I'm funneling into that, looking at the quinone site and the hydrogenase site. And there is sequence homology there, sequence similarity. There's key differences. And you know, when I showed you that nickel iron site, that's a site that's bound to the protein through cysteine ligands, okay? So these are cysteines. So cysteine thiols, in two places, they bridge this metal site. So this is nickel and iron. These are bridging thiolates, and these are terminal thiolates to the nickel. So the nickel has this tetrahedral coordination, but 4 cysteines. And then you have a more octahedral coordination of the iron from these 2 bridging thiolates, water, and these diatomic ligands. Okay, so this is just a sequence comparison in that region of hydrogenase, and let's just compare these. This is an intermediate thing that I'm not going to be able to talk about, but we got the MBH, which has cysteines here that bind this metal site, and now we're here down to NuO. So NuO doesn't have these cysteines, so it's in the process of evolution, the cysteines have been swapped for other amino acid residues. And there's other key differences here. For example, in NuO, one of the defining amino acid residues in NuO is a tryptophan that interacts and supports the binding, or a tyrosine, sorry, a tyrosine that supports the binding of the quinone, and that's present right here, next to one of the residues that used to be there. So what we're talking about here is just kind of the key features in the remodeling of that active site that had this metal center that reduced protons to hydrogen that now has been remodeled to bind ubiquinone. So ubiquinone, if I can get, I'll go with this first, is actually a lot bigger than protons. Actually, most everything's bigger than protons. But this is quite a bit bigger, and so we're talking about a large remodeling of that site. And so what happens here is that the cysteines are actually lost, and the site's remodeled, and this is showing, again, kind of funneling in on the residues, the cysteine, the analogous residues in NuO from the cysteines in the MBH. So they're, all the cysteine ligands that bind that are lost, have different amino acids. The region's modified such that, and I'll cut to the chase, that instead of this tight binding site to this nickel iron center, we have this open site. Okay, so the site, we lose the cysteines, we lose the metal center, the site opens up over evolution to bind quinone to do the reduction of quinone to quinol. So we had, we started with this enzyme that had the features to direct electron transfer to a site of reduction. What evolution did was just remodel that site of reduction from a site that reduced protons to hydrogen to a site that reduced quinone to quinol. So in that process, evolution really, was kind of facilitated by, they have this metal center. If you start to get rid of the cysteines, it won't bind the metal center, and you've got a cavity that then can maybe even more easily be remodeled to a site that binds something big like ubiquinone. Okay, so moving along, that really, I started out talking about the simple example of a hydrogenated electron disulfide Vibrio. This example that was sold by the Fonseca Camps group, he's like one of my big competitors, so I got to remember every time I present, so I have to say his name, his structure. From the Fonseca Camps group, this is kind of a simple version. This is part of this. This is what I just showed you from Pyrococcus. This is part of this big membrane complex. And so this is an example of group 1. This is an example of group 4. We've really most recently gotten interested in group 3. And amongst the enzymes in group 3 are these electron bifurcating or confricating hydrogenases. And that's really the topic of our Betsy Center. Okay? One of the major topics in that, one of the major thrusts, and probably our biggest thrust area, is this electron bifurcation. So let me explain to you what electron bifurcation is. So this is our team, and we met as a team here in this room. This is this room here. So I brought everybody to campus here. Let's get to the meat of this. This electron bifurcation or confrication. So this is what is being billed as the third energy conservation mechanism. Okay, we got substrate level phosphorylation. I showed you what that was. Electron transfer phosphorylation. This electron bifurcation or confrication is a third mechanism. And so what these enzymes do, they either bifurcate the reducing equivalents at one redox potential to generate reducing equivalents at different potentials, or they confricate reducing equivalents at two potentials funneling in, and that's the confrication example I'm showing here, that result in an intermediate potential. And so what this does, like in the context of biofuels, one of the major problems with reduced products like biofuels is a lot of the energy metabolism is directed to forming electrons at the potential of NADH. And that's not really a low enough potential to form some of the reduced products that are most relevant to biofuels. What these bifurcating mechanisms do is allow you to take some of the electrons from NADH, which aren't really sufficient to produce biofuels, but mix them with other electrons in metabolism, like from ferredoxin, to have a lower midpoint potential. Mix them up, and then they spit out electrons that are at an intermediate potential that allow the reduction of things that NADH would normally not allow. So really what we're talking about is, in the context of biofuels, being able to tap in to the pyridine nucleotide pool for producing reduced products, which could be huge. This is just kind of an impact on some of the yields in metabolic pathways, and that Mike gave me. In fact, for all metabolism, these bifurcating mechanisms, because they allow you to sort of balance the electrons better, you can get better yields of ATP and reduced products in the presence of these bifurcating mechanisms. The main things that make a bifurcating center and really the thrust of our project is to try to understand how this center works and how it's gated. Because actually, if you look at this part, this is favorable. This will go. And so it doesn't need to use these. And so really what this center allows you to do is get these in the mix. And so it's got to gate the electrons. You get some of the electrons here, but stop until it gets electrons from NADH to go to reduced products. And so that's really the holy grail with understanding these processes. There's another example here, and this is, so I showed you kind of in the complicating mode, right? You got two things that make one reduced product. They also work in a bifurcating mode, and this is a really nice example from nitrogen fixation. And nitrogen fixation, when you're doing nitrogen fixation, it requires a lot of ATP and a lot of reducing equivalents. And so balancing the ATP and reducing equivalents is really critical for doing biological nitrogen fixation. What this enzyme called Fix does, it's a bifurcating enzyme that takes electrons from NADH and then bifurcates those electrons. it transfers half of the electrons to ubiquinone, which has a redox potential of about plus 100. And by doing that, it sort of concentrates the rest of the potential so that it's low enough potential to reduce this Fix-X, which is a specific iron-sulfur protein that can then funnel electrons to the reduction of nitrogen to ammonia. Typically, NADH isn't at a low enough reducing potential to participate in nitrogen fixation, reduce nitrogen to ammonia. But by doing this bifurcating reaction, we get half of the electrons from NADH go to a higher potential, half to a lower potential. And if you think about this in the context of the electron transport chain, this is really just kind of stealing away the first step of the electron transport chain to then be able to get reducing equivalents, okay? So you're losing some of the ATP you would make in the electron transport chain and directing that towards reducing equivalents. And so this is how nitrogen-fixing organisms can then balance the ATP and reducing equivalent needs of this reaction, biological nitrogen fixation, reducing nitrogen to ammonia. It takes 8 electrons, a lot of reducing equivalents. It takes a ton of ATP here. I have this is kind of a poor example here because I'm taking 1 ATP and making 16 magnesium ADP, which doesn't really work. It's not balanced, but there should be a 16 here as well. It takes a ton of ATP. And so balancing that with, for example, in an aerobe, and in that slide I said that, Fix is really a property that correlates more with aerobes. So balancing that electron flow and how much electrons you partition through respiration and the production of ATP with electrons that then can be used for this process is very critical for the optimization of metabolism under diastrophic growth conditions. Okay, so let me talk about this nitrogen, and I'll end here, and I think I just have about 5 more minutes of stuff on nitrogen fixation. So this is another one of these, we work on these enzymes that they have funky, complex metal clusters. This is another one that has funky, complex metal clusters, and really getting nitrogen reduced to ammonia, that's the highest activation barrier of any biological reaction. You've got a nitrogen-nitrogen triple. And to get over that activation barrier, you need a really funky metal cluster, and I'm not going to really talk about the details of that, pumped full of a bunch of electrons. And so you need a pretty complicated apparatus. The process occurs in a wide range of microbes, both anaerobic and aerobic microbes, but it's an oxygen-sensitive enzyme. And organisms have a variety of different mechanisms, some that we're familiar with, to protect the enzyme from oxygen. Some just do it under anaerobic conditions. Some, like the organism we work on, it's an aerobe, but it respires at a really high rate, so the interior of the organism stays fairly reducing. But we know these things like heterocysts in cyanobacteria, those are little cells, types that create an anaerobic environment, or like root nodules, that's a symbiotic relationship with a plant that creates a low oxygen tension environment to conduct nitrogen fixation. So I showed you this metabolism in Clostridium pasturionum. We did some studies in Clostridium pasturionum looking at the expression of different hydrogenases. And so Clostridium pasturionum has 4 different hydrogenases. And so they do reversible hydrogen oxidation. They can either produce hydrogen from protons, or they can oxidize hydrogen to produce electrons. And so in this case, I was talking about the role of CP1 in reoxidizing or recycling reduced electron carriers. What we see is if we make a transition to diazotrophic conditions, that's really downregulated. We have another analogous iron-iron hydrogenase, CP2, that's upregulated. And so what's going on there is a shift in metabolism. Now, we're under diazotrophic conditions, we need all the electrons, right? We don't want this recycling of electron carriers, we don't have that problem. We need to funnel all the reducing equivalents to the reduction of nitrogen to ammonia. And so you sort of turn off this metabolism and then divert all the reduced ferredoxin to nitrogenase. So I've got nitrogenase here now to reduce nitrogen to ammonia. But this reaction, it actually produces ammonia and hydrogen. And so this CP2 is actually involved in recapturing the hydrogen that can then be used to reduce nitrogen to ammonia. So it's really careful about the electrons. It's trying to keep all the electrons and direct them towards nitrogenase. And so really, nitrogenase, we turn off the CP1 because, this was a problem, accumulating the reduced ferredoxin. But nitrogenase here solves the problem. We don't need CP1 anymore because nitrogenase solves the problem. It can recycle the reduced electron carriers. We have this other one that recaptures that little bit of hydrogen that's kind of like a waste product in nitrogen fixation. So Fix, we think, is key in the balancing of electron and is again associated with aerobic nitrogen fixation. So I just want to finish with another point about this aerobe. So, in nitrogen fixation in aerobes, it's more complicated, but there's more available energy. And so what we've been looking at recently is the differences in the suites of genes in anaerobes and aerobes that fix nitrogen. Okay, and so here's just one example here, and I just have a couple more slides. So this is an extreme example. Okay, so this is Methanococcus. This is a methanogen. Methanogens are really the only archaea that can fix nitrogen. Very simple gene cluster. 1, 2, 3, 4, 5, 6, 7 that I have noted here. But for Azotobacter vinlandii, our aerobe model organism, a much larger suite of genes. Now, the suite of genes, one might think, okay, well, it's oxygen sensitive. Those are all directed at protecting the enzyme from oxygen. But that's not the case. These genes are really not involved in oxygen protection. That's evolved independently, and that's these things like heterocysts or respiratory, facultative anaerobes. We've got temporal sort of regulation of nitrogen fixation, like in single-cell cyanobacteria that only fix nitrogen at night when it's anaerobic. Those are independent mechanisms. So what are all these genes and why do we care? And this is really part of the basis for our synthetic symbiosis project. What we'd really like to do here is to figure out how to transfer nitrogen fixation and allow nitrogen fixation to impact a larger scope of crop plants. So, I mentioned symbiotic nitrogen fixation. This relationship with leguminous plants. They can actually benefit from nitrogen-fixing organisms. They create root nodules and environment, and the plants feed the microbes carbon, the microbes feed the plants nitrogen. And so we'd like to have that relationship be applicable to a much larger scope of plants, and that's a project that we've initiated this multi-investigator project from NSF with a synthetic biologist. I had this guy for a seminar, a pretty quirky guy, Chris Voigt from MIT. And what he does is, I don't think he knows anything about metabolism really. What he knows how to do is how to take apart operons and just reconstruct them using synthetic promoters, terminators, ribosome binding sites. The whole field of synthetic biology. Can we apply that to this problem? And so that's really part of this synthetic symbiosis project, this NSF-supported project in the lab. We use our knowledge of the determinants of nitrogen fixation and synthetic biology to design tailored symbiotic relationships with important plant crops like cereals. This is an example of a cereal, or even maize. So that's a partnership. I mentioned this is Phil Poole in the UK. This is Chris. This is Jean-Michel N. at Madison, and Michael Edvardi at the Nobel Foundation, and this other guy, Giles Olderoyd, at the John Ennis Center, which is a big plant biotechnology center in the UK, or agricultural biotechnology center. So our goal there is to combine the desirable properties of a well-known organism that naturally colonizes the rhizosphere, this Pseudomonas species, Pseudomonas protogens, PS5. And then adapt that to fix nitrogen, use the nitrogen fixation genes and the nitrogen fixation capabilities of our model organism, Azotobacter vinlandii, which is a robust nitrogen fixer. It uses the energy from an aerobic metabolism to fix nitrogen at a high rate. And so we'll have robust nitrogen fixers that will naturally colonize the root systems of a wide variety of plants. Okay, so that's in simple terms, that's what the project's directed. So one of the things we're doing in the beginning of that is just trying to figure out what suites of genes we should be doing synthetic biology programs. So we're informing Chris's studies. And so we really wanted to understand better the diversity of genes. And this is my last slide, fairly complicated slide. And this is really work done by Eric Boyd, and Amaya's been working on this project as well, really cataloging all the nitrogen fixation specific genes in available genomes and correlating them as a function of their nitrogen fixation phylogeny. So what he did was take the structural genes, or the genes that encode the actual enzyme. So it's H, D, and K. There's 3 genes that encode the actual enzyme nitrogenase. And then there's all these other genes that are involved in sort of building these cofactors and some regulation. Okay? So he took all the complements of genes and superimposed them on the HDK dependent phylogeny. Okay? So what he has in black is the simplest. In black shows if they have the genes in it. Simplest gene clusters are in anaerobes. It's more complex in aerobes. That's shown here in green. We have 2 gene products, I1 and I2, which are post-translational regulators. Okay? They just kind of interact with the protein and it doesn't. And then we have A and L here are transcriptional regulators. So you see A is associated with a lot of aerobes, I and I1 are associated with anaerobes. And so I'll just quickly go through the conclusions of that, which is really the first generation information for Chris's work. So in anaerobes, they have a smaller complement of genes and have post-translational regulation. And so we think that's really reflective of an anaerobic lifestyle. You know, a little bit of energy, not much big transitions in metabolism, protein turnover much lower in anaerobes because you don't want to be making proteins over and over and over again with that sort of energy metabolism. Aerobic metabolism, larger complement of genes, and transcriptional regulation. And so what we think that means is that anaerobes or aerobes in that lifestyle, a lot of transition in metabolism, a lot more energy. It's transcriptional regulation, so it can turn off nitrogenase that way. It's turned off that way by virtue of the higher protein turnover rate. So the proteins are turned over higher, and you need this larger suite of genes just because you need that maturation process, making all those complicated cofactors, to happen much more efficiently. If you make it sort of once and just keep it around, then you don't need that process to be efficient. If you're making it over and over and over again because your regulation is at the transcriptional level, you need to be able to make it much faster. Okay? So if we want this to work in aerobes, we need all these genes. That's what we tell Chris. Okay, so this is a picture of my lab with one of our yearly activities. We're actually doing this tonight. This is last year's. So we're doing this tonight in the lab. We get together and drink a lot of beer and color pumpkins. This is, I've already mentioned Mike, but this is the people of his group that's involved, and Eric's had a really special role on this project. And I talked about Amaya and Florence are really the ones that are working on the synthetic symbiosis, and Oleg was really responsible for the structural stuff. Sorry, I went a little bit over, but that's it.
So some of you may have to go, that's fine, and the rest of us can stay and ask questions.
On the fixed story, it's very interesting. I noticed on your side you had another component of 6C.
Yeah.
So is this guy necessary for the bifurcation?
So fix A and B are the part that are that's oxidizing NADH.
Right.
Then that also has the bifurcated center in it, we think.
Okay.
Has the flavin center.
Okay.
But C has the quinoline.
Ah, okay. So
And
that's
it
the relay.
has a little greasy patch, too.
Okay, so that's the relay, that's one of the relay points, right, of the bifurcation? Huh.
Well, somehow you get that reduced there, and then it's gated. It's really got a, so it's gated for the transfer to C, because,
I
that's
see,
favorable.
yeah.
You can
Okay.
just run
So
electrons
it's
all day
gated
long.
At the A and B. The
Yeah.
gating is at the A and B site, not
Yeah.
at the C site, right?
Yeah, so that and we don't know if, that's gated and then you get a transfer to the ferredoxin and then reduce quinol, or that's some kind of concerted thing. Really not much known about the biochemistry of these.
Really interesting.
I think that the discovery of what these enzymes are potentially doing is only maybe 4 or 5 years
To go back to your bifurcation slide, I was a little puzzled by one thing.
old. Uh-oh.
Quite a ways back,
It's got to be after this stuff. Which, the,
What was the first one you're
this
talking-
thing?
Yeah, yeah. I was puzzled what the carbon dioxide is doing there. I understand everything else except the carbon dioxide.
Well, I
and
didn't talk about that, but, with DOE's not just interested in reduced protons, they want reduced carbon, and we've got some mechanisms where we can reduce carbon. And that's not really showing that here.
Yeah,
But it's
so that's
reduced
by
carbon with
carbon.
things like methane, ethane, propane, those types of things. Is
So here you're just adding water to the CO2 to get bicarbonate.
Right.
Yeah.
So that's,
So I thought maybe, okay.
yeah. It's really supposed to more show we can get CO2 to reduce products. And actually one person on the team, this guy at Utah State, this discovery that you can trick nitrogenase, you can make site-directed mutants, and it'll actually reduce CO2 to things like methane, and if you use precursors, you can use maybe like ethylene and CO2 and make propane and things like that. So the DOE is really interested in that, and that's I think we're missing something in the slide here. I'm not surprised that you're the one that.
About the timing, how long does the electron have to wait in that center
Yeah.
for the
No.
NADH?
You mean
Bifurcating center?
Center, yeah, so when the electron goes from flavin to the center, right, to the redox center, how long does it have to wait for NADH? It's just by chance?
well, presumably, presumably that's what the center does, is coordinates all this, okay? And so it's not going to reduce anything. It's got a gating mechanism that says, okay, I'm not going to reduce anything until I at least get some of the electrons from this, okay? So that's what happens. I have no idea how long it takes, and I have no idea how that works. That's what we have, like $10 million in 4 years to figure that out.
Is that a bit like the cytochrome C oxidase complex 4, where 4 electrons have to be transferred before you can actually reduce, but the electrons have to wait, right? It doesn't get transferred to the oxygen until somehow the complex has this. So maybe is that kind of a new design based on cytochrome C oxidase?
Yeah, there could be a relationship between.
I mean, I don't know.
And also the location of flavin and NADH is different, right? So they have to come from different compartments or not?
What do you mean that?
I mean, if you produce NADH from glycolysis, right, where that happens and where you get the flavin, wouldn't that, like the timing would be, I don't know.
Yeah.
I'll ask you 4 years later.
Yeah, there you go. Stay tuned.
More questions? Okay, well, great. Thanks, John.